Welcome to the video on Nitrosamine and NDSRI impurities. So in this video, the key considerations for Nitrosamine and NDSRI's impurities will be discussed in detail. I have referred EMA guideline, US FDA guideline, Swiss Medic guideline, Brazil guideline for making this video. Also, I have referred some of the literature which are available uh, on the internet and some case studies I have referred. So I welcome all the viewers for this video and I hope you will get much benefit out of this video. So let's start with the basics quickly. Nitrosamines are the chemicals which are considered as a probable human carcinogens based on the animal studies, and these are the cohort of concern. If you go to ICH M7, you will get in detail information about the nitrosamines. These are the compounds which are impurities and these contain the nitroso group. So basics we will not cover in detail, only as a revision type we will see. Nitrosamine impurities are known to be mutagenic and carcinogenic, and that's why nowadays nitrosamine is a very important topic to understand. The nitrosamine impurities have very stringent limits because these are potent, and very small exposure of these impurities can lead to cancer and tumors. These impurities may be formed and get incorporated into the drug substances or the drug products through the different ways like reagents, catalysts, solvents, raw materials, APIs, and used into the formulation. Also, these are formed during the manufacturing process. The US FDA and other regulatory authorities have enlisted the nitrosamine examples like NDMA, NMBA, NMBA, NEIPA, NDIPA, NDPA, NMPA, NDBA, and others. So these are also called as small nitrosamines because these are not related to the API structure. These are the small nitrosamine type of compounds, and these are the examples for small nitrosamine impurities. Another class is nitrosamine drug substance related impurities, commonly called as NDSRIs, and these are the nitrosamine impurities which have the similar structure like the drug substance or the active pharmaceutical ingredient. NDSRI is a category of nitrosamine that are structurally like the active pharmaceutical ingredient, that is drug substance in the drug product. NDSRIs can be formed during the synthesis of API, during the manufacturing of API, and also during the manufacturing of drug product. These are also may get formed during the shelf life or storage period or stability period of the drug product. These substances differ from the small molecule nitrosamine impurities as these are related to the drug substance structure. So these are quite big in molecular weight compared to the small nitrosamines. The examples here I have enlisted like N-nitrosopropanol, N-nitrosoquinapril, N-nitrosoabacavir, N-nitrosoatenolol, and many examples are available in the literature. You can go through those literature and find the examples. So I have made a video on nitrosamine NDSRI impurity basics and limits. So video is available onto the Pharma Learning in-depth channel. You can visit that video. It is quite long video, and in that video you will get all the basic information. So here I am not including that much information about the basics. Now we will see the key considerations. So first key consideration is the raw material. What is the role of raw material? How the raw material can play a role in the NDSRI and impurities. So as we know that the nitrosamine formation is triggered by some favorable conditions like nitrite presence, or NDSRI formation largely depends on to the nitrite presence. Also acidic condition is there and some contamination is there. So these conditions increase the chances of NDSRI formation, and also the small impurities get formed. So important consideration here is to study and select the excipients with low nitrite contents. Example is nowadays microcrystalline cellulose is available in the low nitrite grade. So that excipient you can select if you have a problem of nitrosamine impurity in your product. If you know that the API is sensitive for NDSRI formation, then it is always recommended and it is always better to select the excipients with low nitrite content. Also some excipients are available with low peroxide content, low formaldehyde content, or the purified excipients or pure grades of excipients are available. So those excipients can be used whenever you face nitrosamine challenge. Test the raw materials for nitrite impurity levels. That is the another key consideration. Whenever you take the material into the batch, that material should be tested for nitrosamine impurity contamination. Also for the nitrite impurities. See, there is no specific limit for the nitrites to be considered for having the low risk of nitrosamine or no risk of the nitrosamine. You cannot say that if I select the low nitrite grade excipient, that time I will not get the nitrosamine impurity or NDSRI impurity. It is not like that. But it is a key consideration whenever there is a problem of nitrosamine or if your product is sensitive for the nitrosamines. Manufacturing process also is considered for a risk assessment, and it is related to the risk of impurity formation. Some API are more sensitive and some are not at all sensitive for the nitrosamine impurity formation. That's why whenever you do the risk assessment and if you found that the API is sensitive, that time go for the selection of raw materials which have the low impurity content. Ask vendor for their risk assessment and declaration for the nitrosamines. Always keep in hand the nitrosamine declaration provided by the vendor. Now another key consideration is the risk assessment. So first of all, during the risk assessment, do the literature search regarding the API, regarding the finished product if you are working on to the generic formulation. Also take the literature from DMF or use DMF as a literature, strong literature base. Also ask the API vendor or API supplier for their literature regarding the nitrosamine impurity for the particular API. Consider the API chemical structure to start the risk assessment. This approach is called as chemistry based approach. So check whether there is a presence of nitrosamine nitroso group or amine functional group, mainly the secondary amine. Presence of nitrosetting agents like sodium nitrite. Presence of favorable reaction condition like acidic pH environment, and the use of high temperature and heat in the process. So first is amine functional group in the API, presence of nitrite impurity also in the API, and the requirement of higher temperature processing condition for making your drug product. Also consider raw materials, manufacturing equipments, any chance of contamination, cross contamination, any manufacturing problem. Also primary packaging materials like nitrocellulose, then use of acidic material into the formulation, pH of the formulation, and the recommended storage conditions. Then identify the risk, and if the risk is there, then you should go for the confirmatory testing. And if it is confirmed from the confirmatory testing or analysis that the the nitrosamine impurities are there, then you have to set the limits as per the acceptable intake limits. So that is called as AI limit, and then based on that experience, you should set the control strategy so that your batch to batch consistency will there and the nitrosamine will be controlled. Now what is confirmatory testing and testing for the nitrosamine impurities? So based on the risk assessment, the requirement of confirmatory testing can be conducted. If risk is not there, then no testing is required, and if a risk is involved, that time confirmatory testing is must. And if you are working on to the generic products, then the literature search and the literature information can give you the very good information and it can help you a lot for making the decision regarding confirmatory testing for nitrosamine impurities and for the NDSRI impurities. So whenever you are going for the limits, setting the limits for nitrosamine impurities, go for the study of FDA guidelines, FDA guidances. One guideline here I have included, that is control of nitrosamine impurities in human drugs. That is a guidance for industry from US FDA. You should study that guideline for giving the or for setting the impurity limits. Here I have included some structures for the small nitrosamine impurities. Now here in this guideline you will get the AI limits for some of the nitrosamine impurities. So here AI limit is given in the ng per day, that is nanogram per day, and this nanogram per day is derived on the basis of maximum daily dose. So directly you can take these limits. Then coming to the limit derivation or deriving the limit, that is as per the US FDA guideline, the maximum daily dose of 880 mg is considered, and based on that the calculation is done for nitrosamine impurities. Also you should consider the ICH M7 guideline for nitrosamine impurities, and as per the ICH M7, the limit of 1.5 microgram per day is given for the nitrosamine and NDSRI impurities. Now NDSRI limits are based on to the predicted carcinogenic potency categorization, that is called as carcinogenic potency categorization approach, that is CPCA based predictions for nitrosamines and the NDSRIs. So here I have included a reference from EMA guideline, question and answer for marketing authorization holders or applicants on CHMP opinion for the article 5 of regulation. So this question and answer document will give you the potency category as 1, 2, 3, 4 and 5. So if your molecule or if your impurity comes into the category 1, then AI limit will be 18 nanogram per day, that is the stringent one. If it is 2, then it will be 100. If it is 3, it will be 400. If it is 4, it will be 1500 nanogram, that is 1.5 microgram. And 5 category will give you the Ng per day limit of 1500 nanogram, that is also the 1.5 microgram. So if you go through my channel, you will find other videos which are related to nitrosamines, and from that video you will get the clear information regarding the limits. Here I have considered only the key considerations, so I am not going into much details. Then another is the FP specification. So if you have the impurity problem, or if you know that the impurity is present into the formulation, then what is the next stage? What should be done? See, the regulatory authorities are different for different markets. The limits are a little bit different for different molecules, for different impurities, and also these are product specific based on to the maximum daily dose and based on to the risk benefit involved for the product. The impurity limits can be given or can be discussed with the regulatory authorities. So always know that whenever you are working on to the nitrosamine impurities, you should have a very strong literature. You should have a very strong analytical method, validated analytical method, and also you should have a control strategy. For now, considering the FP specifications, the NDSRI limit, when it is NDSRI or nitrosamine limit, when it is required to be given into the specification and when it is not required, that we will discuss. So based on the risk assessment and the confirmatory testing, if the testing of the manufacturing batches is done and if you find the results are consistently below 10% of the specification limit, then possibly there is no need to include the impurity into the specification and also testing is not required. So if the results are in between 10% to 30% of the AI, that is acceptable limit, then you can go for the skip testing. And if you get the results of nitrosamine or NDSRI limit consistently above 30%, but below the specification limit, that is below the AI limit, then regular testing is not required. If the results are above 30% but within the specification limit, then the testing is required. So 10%, then 10 to 30%, and above 30% is the criteria for consideration of FP specification limit for nitrosamine and NDSRI impurity. For example, if you get the NDSRI impurity 50% of the AI limit, then you should include the limit in the specification and every batch is required to be tested for that impurity. If you get the results consistently below 10%, then there is no requirement of testing, and that's why there should be no requirement for inclusion of the nitrosamine impurity into the FP specification. So based on the requirement of testing, the FP specification can be done. Then consideration for the stability study. If you get the nitrosamine impurity at initial stage, so there are chances that the impurity will be same throughout the shelf life or it may increase. So there are chances that the stability period will have an impact onto the impurity level. The NDSRI may or may not increase on storage and during the stability. So it depends on to the API and the product composition. For some of the APIs or some of the composition, it will increase, or for some of the products, it will not increase. So it is always better to do testing for stability samples. In the research phase of the product development, different packs should be there so that you will know the impact of packaging onto the impurity levels. It is also important to select the composition and process to control the impurity formation on storage. and this is only regarding the nitrosamine impurity. If you are considering other impurities also, that time as a general consideration, the formulation composition and the packaging materials are studied. But for nitrosamines, it becomes very critical because for nitrosamines, the acceptable intake limits are very stringent. For example, I have considered impurity generation on stability for batches made with normal grade of raw materials and the batches made with the low nitrite grade of raw materials. So in the research phase, you can do those exercises, and based on that, if the impurity is getting increased during stability, that time you have to include the impurity limit into the specification. And if it is increasing in the stability period, that time it is must to check that impurity in the initial stage, whether it is below 10%, whether it is below 30%, or whether it is above 30%, then it will not matter because that impurity is getting increased into the stability period. That's why it will become must to include the impurity into the specification. And at that time, each and every batch is required to be tested for nitrosamine. Another is the yellow Q. As the limit for nitrosamine impurities are very stringent, yellow Q becomes very important, and limit of quantitation should be below or equal to 10% of the specification limit. Also, if the impurity is below 10% of the specification limit, then the testing can be omitted. So you can link this thing with the skip testing or routine testing or omission of the testing. Then if below 10% is not possible, then try to get it below 30% of the AI limit, so that you will get the skip testing criteria in your product. So this is regarding the yellow Q. Then higher NDSRI impurity level or NDSRI higher than the AI limit. So if you have a product with higher NDSRI level than the AI limit, so what you will do? So take the steps to reduce the impurity level below AI. That is the first requirement. Then you should consider whether the product is commercialized, whether it is under approval stage, or whether it is approved, or whether it is under the research and development phase. The another step is to contact and inform the respective regulatory body and get the clarity or suggestions based on to the product stage, like under development, approved, or commercial. Perform the studies to establish the safety data for proposal of the limit higher than the AI limit. That means you have to perform the toxicity studies to widen the acceptance criteria. Then how to control the nitrosamine impurities and NDSRIs? It is very important nowadays to have the information for controlling the impurities. Control onto the raw materials and processing conditions. Understand how these impurities are being formed, and based on that, you should make a mitigation strategy. Understanding the pathways for impurity generation is very important. Identifying where the nitrosation reaction or nitrosation reaction is happening or occurring, and mitigating it to eliminate it or to minimize it. Testing the stability samples to understand the impact of storage conditions and the packaging. Also, many of the time people change the raw material source or raw material grade. Sometimes the API source can also be changed, as the different API suppliers will give you the material with different route of synthesis. So based on that also you can have the control onto the NDSRI impurities and nitrosamine impurities. So change in API source, route of synthesis, API from or other critical attributes. Like a crystalline material may be having more stability considered compared to amorphous API, or depending on the particle size or route of synthesis, it may have impact onto the nitrosamine impurity formation. So these are some of the things which are required to be studied. So this is regarding the nitrosamine and NDSRI impurity key considerations, and I hope I have considered the critical points, and I also consider that you got the good information out of this video. Other videos are also available onto the Pharma Learning in Depth channel for the nitrosamine and NDSR impurities. You can go through those videos and you can get the good information from those videos. I request you to share the information to your friends and colleagues, and also to uh, like, subscribe this channel to have more information regarding these important topics. Nowadays, regarding the NDSRIs and nitrosamine impurities, questions are being asked in almost all the interviews. So these type of videos will help you to get the clarity on the topic. Thank you for watching the video.